Hello everyone, my name is Atia Rasheed. I'm a PhD candidate at the College of Medicine and Health Sciences UAEU. I'm truly honored to present my research here entitled Impact of Vitamin D on Acute EBV Infection in a Novel Rapid Model. Firstly, I would like to thanks to the organizing committee of BSI for their kind invitation to speak at this prestigious conference. Before we dive into the details of the study, I will briefly introduce you about the topic. Then I will discuss about the objectives, material and methodology, results, and I will end my talk with the conclusion from the study. Multiple sclerosis is a chronic autoimmune disease of the central nervous system that leads to demyelination, neurodegeneration, and progressive disability of a person. The disease etiology is very complex and not well known. However, several genetic factors such as HLA-DR15 and environmental factors that include infection with different pathogens, lifestyle factors, exposure to the sunlight, and low level of vitamin D are involved in MS pathogenesis. Of the environmental factors, Epstein-Barr virus and low level of the vitamin D are important risk factors for MS development. Epstein-Barr virus or EBV is a human gamma herpes virus that infects the B cells and persists in the form of latent infection during the life of the host. Virus can persist in the form of different type of latencies depending on the type of the infected cells and its gene expression pattern. Involvement of EBV in MS is suggested by the elevated level of serum antibodies against the Epstein-Barr virus nuclear antigen, particularly EBNA1, and also the presence of virus in the demyelinated lesions in most of the MS cases. Despite the presence of virus in MS, the exact mechanism for its pathogenesis in MS is still unclear. Another important risk factor is vitamin D, which is a multifunctional hormone that can either obtain through dietary intake or through UVB radiation from sunlight. Vitamin D is well known for its immunomodulatory role as it maintains or it regulates the immune system by the downregulation of pro-inflammatory immune cells and upregulation of anti-inflammatory immune cells such as B regulatory cells and T regulatory cells. Involvement of vitamin D in MS is suggested by the studies that found higher prevalence of MS in individuals living at high latitude and also having low level of vitamin D. Despite several independent studies on these factors, the interplay between vitamin D and EBV is still unclear. So, aim of this study is to investigate the impact of vitamin D on EBV infection in the peripheral compartment. To address this novel question, we have used or recently established a rabbit model of EBV that mimics the natural human infection. The study was conducted on 39 New Zealand white rabbits. All of the animals were divided into two major groups, vitamin D negative group and a vitamin D positive group. Both of these groups were further subdivided into two subgroups, EBV negative group and EBV positive group. Both of the EBV positive groups were injected with 1 ml of fresh cell culture derived virus through the marginal ear vein of the rabbit. On the same day, vitamin D negative group was fed with the vitamin D deficient diet. On the other hand, vitamin D positive group was fed with the vitamin D positive diet as well as oral vitamin D supplementation every second day for 2 weeks. Blood was collected as pre-treatment blood at day 0. at week 1 and at week 2 at the time of euthanasia. 15 days post infection, all the animals from all the groups were euthanized. Whole blood was collected, followed by the separation of PBMCs and plasma. All of the major organs were harvested, including spleen, brain, liver, lungs, and kidney, and were stored for the downstream analysis. Following the animal work, level of the vitamin D was assessed by using the chemiluminescent assay. qPCR was done to quantify the EBV load in peripheral compartments. In situ hybridization was done to localize the EBV infected cells in different tissues, and immunohistochemistry was performed to check the gene expression level of the virus. Moving towards the results, histopathological analysis revealed substantial differences between the two groups. As we can see here, spleen from the vitamin D positive group is distinct in terms of color and its shape as compared to the vitamin D negative group and its control group, which gives an initial indication that there is something going on between the level of the vitamin D and EBV infection. Plasma level of vitamin D was checked in all of the groups and compared with the pre-treatment samples from all the animals. As expected, level of the vitamin D was decreased in the vitamin D deficient group, and vitamin D level was significantly increased in the vitamin D supplemented group relative to the pre-treatment samples. EBV load was quantified in the peripheral blood mononuclear cells for all the animals and compared with the pre-treatment samples. Surprisingly, EBV load was higher in the vitamin D positive group as compared to the vitamin D negative group. Similarly, viral load was significantly higher in the vitamin D positive group in spleen as compared to the vitamin D negative group and its respective control group, which indicates that level of the vitamin D is having an impact on EBV infection. In situ hybridization was done to localize the EBV infected cells in the formalin fixed paraffin embedded spleen sections. Clinical sample was used as a positive control. Vitamin D negative group spleen was observed with a limited and scattered number of positive cells. In contrast, a vitamin D positive group spleen was observed with the widespread distribution of EBV positive cells, as we can see in this picture. Furthermore, for the viral protein expression analysis, immunohistochemistry was performed, and both of the groups were observed with different level of gene expression pattern. Clinical sample was used as a positive control. Vitamin D positive group was observed with the widespread and higher level of viral latent membrane protein as compared to the vitamin D negative group. Taken together, all of these experiments indicate higher level of EBV infection in the vitamin D positive group than the vitamin D negative group. So, our data indicates a significant positive correlation between the level of the vitamin D and EBV load. In conclusion, in contrast to what has been suggested, we show for the first time in a controlled manner that vitamin D level correlates positively with the EBV infection. These observations reflect the complex relation between EBV and vitamin D, which could potentially lead to the development of new strategies to control the EBV infection in MS pathogenesis. Thank you so much.